Our next topic is meiosis, and you will use this presentation in order to help you with the lab in your lab manual called the Meiosis Lab. So let's start off by saying most organisms reproduce by a process called sexual reproduction. First, a type of cell division, which we called meiosis, has to occur in order to make special cells that we call gametes. When the gametes combine during fertilization, sexual reproduction has occurred. Let's make sure that we understand that mitosis results in asexual reproduction of cells. And these cells tend to be clone-like cells. Okay, let's do a quick review of sexual reproduction. This is a review of what we learned about from chapter 10. We're going to use humans as an example. So here we have our sperm gamete, which please recall that sperm cells are a haploid type of cell. And we're just simply going to do a review of what we talked about at the beginning of chapter 10. It looks like my sperm gamete has 2 chromosomes. That's not human though. So remember, sorry, this should be green. This should be all green. Okay. So if we were talking about humans, there would be 23 chromosomes in the sperm gamete, but I am not going to draw 23 chromosomes. So here also is our egg gamete, not drawn to scale. And this is also a haploid type of cell. And remember, haploid can be symbolized with a lowercase n. We'll use some different color markers to indicate different sequences of DNA. So remember when the sperm and the egg combine during fertilization, okay, the chromosomes will also combine in the fertilized egg, which we call the zygote. Just doing a quick review. The list the paternal chromosomes that came from the sperm cell first. And we'll put those chromosomes together and form our homologous pairs in our fertilized egg, just so we can understand what homologous pairs are. So here's a homologous pair, and here's a homologous pair. It looks like these chromosomes in this homologous pair are much longer in size compared to these chromosomes. There are two homologous pairs shown in this picture. Remember, humans have 23 homologous pairs of chromosomes, but I'm certainly not going to draw all of that. So don't forget that we call these chromosomes, which the egg has inherited, the green and the purple chromosomes, those are called the paternal homologs. And then the other chromosomes that were inherited from the egg gamete are called the maternal homologs. So it appears that each pair is made of one paternal and one maternal homolog. And that forms a homologous pair of chromosomes. Going to move on to the next page. We're going to look at a picture from your online textbook. Okay, so this picture is in chapter 11 in your online textbook. So one of the major goals of meiosis is to reduce the chromosome number by half. I will explain this. The process of meiosis is going to have to go through 2 rounds of cell division in order to reduce the chromosomal number by half in the cells that are produced at the end of the meiosis process. These 2 cell divisions are called meiosis 1 and meiosis 2. So here we have our fertilized egg. And then our fertilized egg goes through G1, S, and G2. So during the S subphase, the DNA replication occurs. Please review chapter 10 notes, which is basically mitosis, if you do not remember the S subphase during interphase. During the S subphase, we took our independent chromosomes and we ended up with duplicated chromosomes, which is what we're seeing here. So this is a duplicated chromosome containing 2 sister chromatids, and this is another duplicated chromosome containing 2 sister chromatids. When the cell goes through meiosis, and that means that the cell is going to either become a sperm cell or an egg cell. Okay, the only type of cells produced during meiosis are gametes specifically, meaning a skin cell, a stomach cell, a blood cell is not produced by meiosis. Those are produced by mitosis. The only type of cells produced at the end of meiosis are gametes. That's the whole goal of meiosis, to make sperm and egg cells. So it's a very specialized type of cell division process in order to make sperm and egg cells. So after the cell goes through interphase, it's going to then enter meiosis 1. The major event to understand during meiosis 1 is that the homologous pairs, remember this is a homologous pair, just like this is a homologous pair, right? So this is a homologous pair. This is an independent homologous pair made of independent DNA molecules, and this is a homologous pair made of duplicated DNA molecules. Why do we have 2 different color homologous pairs? The reason that we have 2 different colors is to indicate the maternal and paternal homologs, just like we did on the last page of our notes. So we'll say paternal homolog, that came from the sperm cell, and that's the maternal homologue. And we're using two different colors to indicate these homologues because mom and dad do not have the same DNA. So the different colors are indicating different sequences of DNA. So basically, our homologous pairs will line up at the center of the cell during meiosis 1, and the homologous pairs will separate. That's going to be the major event that occurs during meiosis 1. Well, one of the major events that occurs during meiosis 1. So after the homologous pairs separate, then the two cells that are produced at the end of meiosis 1 will go through the second round of cell division, which is called meiosis 2. And the sister chromatids will separate. Bring this up a little bit so we can see. So let's go ahead and note that at the end of the process, there are 4 haploid cells produced. And remember that these cells are called gametes, sperm and egg, for example. Now, if we're using color as an indication of the DNA molecule, I want you to notice something about the 4 cells that are produced. In this cell, we have a blue chromosome with a purple tip. In this cell, we have just a plain blue chromosome. In this cell, we have a purple chromosome with a blue tip. And in this cell, we have a plain purple chromosome. So those little color exchanges that we see, okay, in our cells indicate that all the 4 cells produced do not have the same sequence of DNA. So please look closely at the cell and use the color combinations to understand that the DNA sequences are not identical. Do any cells produced by the end of meiosis, and we're talking about these cells here, do any cells produced by meiosis have identical DNA sequences? So the way that you can look at it is you look at the colors of the chromosomes. This chromosome is blue with a purple tip. This chromosome is all blue. This chromosome is purple with a blue tip, and this chromosome is all purple. So all of these 4 cells at the very end, based on the color of the chromosomes, are not identical. If you have to stop or pause the presentation to take a closer look, okay, at the 4 cells that are produced by the end of meiosis, that ensure that they all have different color combinations within their DNA molecules, because these are the DNA, here's 1 chromosome in each cell, then please make sure to study the picture, because you have to understand that meiosis does not make a clone-like cell. Meiosis makes cells that are genetically different, and that means they all have different DNA sequences within their chromosomes. So what we're going to do is go over some key events of meiosis. We are now on question 4. Some key events is that meiosis is a prerequisite for sexual reproduction, meaning you need meiosis to allow for sexual reproduction. In humans, without meiosis, there will be no sexual reproduction. Why is that? Because meiosis makes the sperm and the egg gametes that allow for sexual reproduction to occur. The types of cells produced by meiosis are called gametes, which we also commonly call sex cells. In humans, the male gametes are called sperm. In human females, sorry, the female gametes are called egg or oocytes. Oocytes is the fancy term for eggs. Let's consider the processes that produce different types of gametes. We're going to call the process to make gametes gametogenesis. There are two types of gametogenesis. In human males, type of gametogenesis is called spermatogenesis. When spermatogenesis occurs, human males make 4 equally sized sperm cells. Please do not forget that meiosis makes 4 daughter cells at the end of the process. Now, in human females, the process of gametogenesis has a fancy name. It's called oogenesis. There's going to be one egg, which is called an oocyte, produced, and 2 or 3 other type of cells called polar bodies produced. Only the eggs are fertilized. There is a weird type of cell called a polar body, which is produced when a female is going through meiosis. The polar bodies are produced in order to reduce the chromosomal number in half, which is a major goal of meiosis. Okay. So where does gametogenesis occur? It will occur in the primary reproductive organs, which are called the gonads. And the female gonad will be the ovaries, and the male gonad will be the testes. The testes and ovaries. All right. Let's go over mitosis in a little bit more detail, make sure you understand a few things. The goal of meiosis is to reduce the number of chromosomes by half in order to produce haploid cells. So I want you to make sure when you hear the term haploid, you always connect it with meiosis. Meiosis is the only type of cell division that is going to make a haploid type of cell. Well, it's the only type of cell division that we're going to talk about in this class that will produce a haploid cell. In humans, the haploid number of chromosomes is going to be 23. And remember, the haploid is symbolized by a lowercase n. So if you see a lowercase n anywhere in your lab manual or your notes, your reference materials, please make sure that is a symbol representing haploid number of chromosomes. So in humans, the haploid number is 23. In fruit flies, their fancy term is Drosophila, the haploid number is 4. Can you determine the diploid number of chromosomes for the fruit fruit flies given that the haploid number is 4. So you would basically say 2, and then N, 2N equals diploid. So equals 2. Up here we said N was 4. And that, your answer is going to be 8. So the diploid number in fruit flies will equal 8. So different species have different diploid numbers. So for example, in humans, the diploid number is 46. Whereas in fruit flies, the diploid number is going to be 8. All right, so the term haploid refers to the set of chromosomes that you have inherited from one parent, and the term diploid refers to the combined set of chromosomes received from both parents. The diploid number of chromosomes for a cell is restored when the sperm fertilizes the egg during fertilization. I'll move this up a little to make sure to get all the notes in here. Okay, all cells produced by meiosis are going to be genetically unique. That means they all have different DNA sequences, unlike mitosis, where the cells produced are genetically identical, right? So meiosis is unique, going to make unique types of cells, whereas mitosis makes identical type of cells. And we end up calling these cells clones. They're very clone-like. If you have ever heard of the term clone, that just means an identical copy. So our last major point here, I think it's our last point, is going to be that in meiosis, there are 2 rounds of cell division, therefore producing 4 daughter cells, whereas mitosis only makes 2 daughter cells. So mitosis is going to be chapter 10, right? And meiosis is going to be chapter 11. So we're on chapter 11 talking about the topics of chapter 11, which are going to help us get through the meiosis labs. Okay. A couple of things to point out. Okay, and this is a figure that's going to be in your online textbook, so refer to chapter 11. So the stages of meiosis 1, this will be like the parent cell, will be prophase 1, metaphase 1, anaphase 1, telophase 1. And the stages of meiosis 2 are going to be prophase 2, metaphase 2, anaphase 2, and telophase 2. All right, so during prophase 1, the homologous pairs of chromosomes get very close to each other. And what happens is they end up swapping equal segments of DNA. And this is how the blue chromosomes and the previous couple of pictures had purple tips, and the purple chromosomes on the previous pictures had blue tips. This event is called crossing over. So an event, which we will discuss in a little bit more detail soon, called crossing over, occurs during prophase 1. Okay, So during metaphase 1, the homologous pairs of chromosomes line up side by side at the center of the cell. And then we see that the homologous pairs of chromosomes are split apart during the next stage, during anaphase 1. I need to move this up a little bit. Oh, got a little ahead of myself here. Let's review each one of these stages. So during prophase 1, okay, that's going to be up here. During prophase 1, homologous pairs of chromosomes get really close together and swap equal segments of DNA. Okay, so over here, a homologous pair of chromosomes is shown for us right here. And there are 3 homologous pairs in this picture. And the homologous pair of chromosomes is made of a purple chromosome and a blue chromosome. Why do we have 2 different color chromosomes within a homologous pair? Please do not forget that homologous pairs of chromosomes are made of a maternal homologue and a paternal homologue. So mom and dad do not have the same DNA, which is why there's a purple and a blue chromosome. That indicates different sequences of DNA. So during prophase 1, an event called crossing over occurs. Crossing over is when a homologous pair of chromosomes get side by side, and then they exchange little bits of segments of DNA with each other. So, for example, the blue and the purple chromosomes will exchange blue and purple segments. Okay, and you can see that demonstrated. Okay, when you see in another cell right here, a purple chromosome with a blue tip. Okay, so when you see that, you can say, oh, a little swapping of the DNA has occurred. And that basically occurs due to crossing over. We will look at that in more detail on the next page. At this point, you want to know the event called crossing over occurs during prophase 1, and it includes the homologous pairs getting really close to each other and swapping equal segments of DNA. You will next want to know that metaphase 1 are when the homologous pairs of chromosomes, the blue chromosome and the purple chromosome, they line up next to each other side by side at the center of the cell. Okay, during anaphase 1, you will want to know that the homologous pairs of chromosomes are separated and pulled to opposite poles of the cell, and 2 cells are produced at the end of meiosis 1 activities. Let's go ahead and look at meiosis 2 activities. So here are the 2 cells that are produced from meiosis 1. Okay, the chromosomes are going to line up at the center during metaphase 1, and the sister chromatids are going to be separated during anaphase 2. So those are just some events that are highlighted, okay, in each of these stages of meiosis. You will want to make sure you understand this because you are going to be building chromosomes and simulating these stages of meiosis in lab. Okay, so key events to note during the process of meiosis will be during prophase 1. Genetic recombination occurs due to an event called crossing over. So we don't have the blue and purple chromosomes anymore. Instead, the online textbook has decided to choose red and blue. Okay, in lab, you will build red and yellow chromosomes. So I know there's a lot of different colors, but the concepts are still here. So we have our homologous pairs lined up. So here's a pair, okay? Why do we have within a pair, this is our pair right here, right? This is our pair. How come within a pair we have one blue chromosome and one red chromosome? Please do not forget the reason we have different color chromosomes is because one is the maternal homologue and the other is the paternal homologue. Do mom and dad have the same DNA? No, they do not, which is why we have blue and red chromosomes. The colors indicate different DNA sequences. Indicate. So during crossing over, the homologous pair of chromosomes swap an equal segment of DNA. So you can see right here, the blue chromosome now has a red tip, and the red chromosome now has a little blue tip. That indicates that crossing over has occurred. So want to go ahead and highlight that crossing over occurs between non-sister chromatids within a homologous pair. So now you have a blue chromosome with a red tip and a red chromosome with a blue tip. Okay, during metaphase 1, we're still talking about the stages of meiosis. An event called independent assortment occurs. And this further shuffles up the DNA. So, for example number 1 or example number 2, meaning that during meiosis, okay, in the testes or the ovaries, when meiosis is occurring and the chromosomes are all lining up at the middle of the cell, the chromosomes can be, basically organized randomly. So, for example, you have your big A chromosome here and your little a chromosome here. This makes up a homologous pair. So, here's a homologous pair. It looks like this cell has two homologous pairs. Here's one homologous pair. Here's the second homologous pair. Okay, so you have your A chromosomes homologous pair, and you also have the B chromosomes homologous pair. Okay. You can line them up in the cell during metaphase with the big A's and the big B's on one side, or you can shuffle that up a little bit in our example 2 or possibility 2 and have the big A's and the little b's on one side. So if you look at the resulting gametes, just due to the placement of the homologous pairs of chromosomes during metaphase 1, okay, you can see that the gametes that are produced are all going to be genetically different. Okay, so the randomness and the alignment, coupled with the crossing over, are responsible for basically genetic variation. Okay, so just some other notes, just a little review from the last couple of pages. Homologous pairs of chromosomes separate during anaphase 1, and sister chromatids separate during anaphase 2. Okay, we are on the last page of our notes. We are almost done. And now I'm just going to review the life cycle of sexually reproducing organisms. And of course, I'm going to use a human as an example. So let's go ahead and draw this diagram again and make sure you guys all understand this. Here's your sperm. Now remember, the sperm is haploid, right? Haploid. Here's the egg. But the egg is also haploid. So they fertilize and we make a zygote. And then the zygote is diploid. Okay. So the zygote then develops into embryo, fetus. Let's just skip some steps. Adults. So we skipped a few steps in growth and development. Okay, but one thing that you want to make sure that you understand is that growth and development and tissue maintenance for example, one maintenance will be repair, are all events that are affected by mitosis. Affected by mitosis. And remember that mitosis is asexual reproduction of cells. The sperm and the egg is produced by meiosis. So meiosis is needed to produce gametes. Which are needed for sexual reproduction. So we want to make sure that you know that both mitosis and meiosis are important in the human life cycle. Why is mitosis important? Without mitosis, we would not have growth and development, right? From zygote to adult, and we would not have tissue maintenance and repair. That is all a product of mitosis, which is asexual reproduction of cells. Without meiosis, we wouldn't be able to make sperm and egg to allow for sexual reproduction. So both meiosis and mitosis are important in the human life cycle or in life cycles. Okay, see you next time.